Welcome to Brain Ponderings, your window in the world of neuroscience research and conversations with experts at the forefront of their field. Today, it's my pleasure to chat with Tessa Montague. She's at Columbia University, where she's a Howard Hughes Medical Institute Hannah Gray Fellow and Department of Neuroscience. She's kind of at a position where she's like independent, but still within a huge laboratory of Richard Axel, Nobel Prize winner, for his work on olfactory system. But anyway, she has a really, I kind of looked into her background. She has a really interesting background. And today she's going to talk about how the brains of octopuses, squids, and cuttlefish, which is what she works on, uh, control camouflage in their skin. It's really a fascinating uh, feature of these creatures, something we can only dream about doing ourselves. So welcome, Tess.
Thank you very much. Happy to be here.
Okay, oh, and by the way, I see she has a website. It's pretty easy, tessamontague.com. And I'll put some links in and maybe she'll give me some links to videos of these
Mhm.
animals in the ocean changing their skin color and patterns and so on in response to where they are or whatever's around them. Uh, okay, so your background, you're from England or at least British Isles somewhere.
Yes, I'm from England. Yeah.
And where did you grow up in England?
I grew up in London, kind of
Uh,
central London. Yeah.
okay, so in the big city.
Mhm.
And when did you first get interested in science and neuroscience?
Um, I think my whole life I was interested in science. So I come from a family of musicians and artists and so forth. There's not a single scientist in my family, but I really enjoyed it in school. And I was quite influenced by the TV shows I was watching. So I mean, at first I wanted to be an astronaut, like many kids. Then I wanted to be a lawyer because I watched Ally McBeal. Then I wanted to be a CIA agent. And I was pretty serious about that. I'm actually half American, so technically I could be a CIA agent. Um, but then to get into the sort of best universities in the UK, ideally you have some kind of summer experience. So I applied for a summer research fellowship when I was 17 just because honestly, it's just because I thought it would be good on my CV. And then I just deeply fell in love with science. So I thought it was incredible that you're basically set loose in the lab to just discover something about the world or the universe that no one knows before. And there's no kind of set of instructions or rules for how you make these discoveries. You have to be kind of creative and think outside the box. And I just thought it was the coolest thing in the world. So
What university was that? A university in London?
it was at what's called the National Institute for Medical Research.
Oh,
It's now been kind of consumed by the Francis Crick Institute. Um, it was on the kind of outskirts of London in a building that they filmed one of the Batman movies in.
oh, cool.
Um, and it was, yeah, it was very cool. I worked with Dr. Malcolm Logan on chick development. And after that summer, I knew all I want to do is be a scientist.
Batman, I thought he was in New York City, Gotham.
I think it was one of the old movies, and probably
Okay,
it was meant to be New York. It's just a
okay,
quite an imposing concrete
okay.
building, so
I see.
good for Hollywood, I guess.
All right, so then you decided to go to college and major or study some area of science, biology, I guess.
Yeah, so yeah, in the UK, you already start deciding what you want to study when you're 16. So I was already doing all
Oh,
sciences and maths, and I chose to study what's called natural sciences at Cambridge University. So actually, it's the broadest program you can get where you study not only biology but also chemistry and some maths. But then you gradually specialize. So I ended up doing my degree in genetics.
oh, nice. And right. So, and did you do research then in a lab while you were
I
taking?
did, but also the difference between the UK and the US is you don't really have an opportunity to work in a lab during the term time, semester time.
Hmm.
So it's just reserved for the final year. You have like a 3 month stint of full-time research. So I continued doing some summer research, and then I worked in a lab, in a Drosophila development lab, um, Daniel St. Johnson's lab at the end. And then I did a 1 year research master's before applying for PhDs.
Yeah, so these, the fruit fly, the Drosophila, the worms, the elegans. I worked on, well, my PhD, I was studying neuroendocrine system that controls molting in crabs.
Ooh, cool.
And then first part of my postdoc lab, I was in the lab of Stan Cater, who was a developmental neurobiologist. And we were studying growth cone behaviors
Mm.
and identified snail neurons,
Oh, wow.
which have, so these snails, and I don't know, squids, I don't know, I haven't looked at a squid brain, but the snails have giant neurons, right? You can see
Mm.
them, some of them with your eye with unaided, but we'd use like a dissecting microscope and suck out individual neurons from their brains and put them in culture and then watch their growth cones grow. And we discovered neurotransmitters regulate their growth and regulate synapse formation. But anyway, my point is these invertebrate models, there's been, I guess I don't know, dozens probably of Nobel Prizes awarded to scientists who discovered something in invertebrates, some of these models I mentioned, that then turns out the genes and the proteins that they discovered there, they're pretty much the same as in us. One of the most recent ones was autophagy, right? Which
Mm.
was actually discovered in yeast. So anyway, uh, your work, which there's not much known about what you're doing actually, from what I can tell from the literature on the control of camouflage. Okay, so then from Cambridge, uh, then you came to the United States.
Yeah, exactly. So I think partly because I knew that I wanted to be a scientist, I wasn't so intimidated by choosing a 6 to 7 year PhD rather than a British 3 year PhD. So I decided to move to the US for this very long uh, training period. And I did my PhD at Harvard University with Alex Shear, working on zebrafish development.
Oh, nice. That's another. Can you quickly say why zebrafish are so valuable?
Yeah,
It,
for sure.
yeah.
Yeah, the reason we study zebrafish or zebrafish, I mean, especially in development, is that the fish lay eggs outside of the body, which doesn't sound particularly meaningful, but obviously, if you want to study how mammalian development happens, you have to somehow watch it happen in utero in a female. So with fish, they lay the eggs in the water and the males deposit sperm and then they fertilize as the eggs fall to the bottom of the tank. So you can just collect as many embryos you want and you can watch them develop. And then they're transparent. So all of the biology happens right before your eyes. And I think this is a theme of biology that I really love, which is being able to see it. And of course, much of biology is very visible, but not everything. And watching a zebrafish embryo develop is kind of magical because within just 24 hours, it goes from a fertilized egg to a beating heart.
Wow.
And so it's incredibly fast, which is also why people study it. It's transparent, it happens in a dish. And so it's quite a convenient system to kind of uncover the basis of how development happens.
And you can also now with technologies like these fluorescent probes for calcium, you can look at activity in real time and in theory in all neurons, but many neurons as you want. And do they do optogenetics? And I guess they do, right? In the
Yeah, they
zoo?
do optogenetics. So yeah, you can control. I mean, I wasn't working on neuroscience back then, but you can
Ah,
control the activity of neurons. You can record or image the activity of neurons. People have labeled all kinds of different genes with fluorescent proteins. So, for instance, I did a lot of outreach work, and we had this line of zebrafish that have fluorescent blood cells. And just putting a kid in front of a microscope and watching these blood
ah.
cells zip around the body and get pumped into the heart and around the brain, like it's pretty magical.
Oh, wow. Yeah, that's really cool.
Yeah.
Huh. Are some of those videos up on the internet somewhere?
I think so, yeah. I should
Okay.
be able to find one. Yeah.
Okay, I'll have to track them down. Maybe if you can send me a link, I'll put it in.
Yeah, definitely.
Mhm. Um, okay, so what was your main discovery in the zebrafish?
I think the main discovery was that we were interested in how embryos form what's called endoderm and mesoderm. So these are when an embryo is just a ball of cells before it's formed organs and so forth, it already starts specializing what certain cells will become. And the endoderm will form your internal organs, the mesoderm will form your muscle and bones. And until this point, everyone thought that the process of deciding certain cells will become endoderm and mesoderm was controlled by a gene called Nodal. And we basically found that there was another gene or protein called VEG1 that this whole time had been there and was forming what's called a heterodimer. So rather than there being just nodal doing this process, it's like a signaling molecule. There was another protein called VEG1 that forms a dimer with it and helps to control this process. So we
And,
kind of, yeah.
and okay, so I don't know about veg when I've heard of nodal. So it's not vascular endothelial growth. Is it,
No.
what's the VEG stand for?
Um, oh my gosh, I'm thinking on it. I mean, it was originally discovered in
You
Xenopus.
already defended your thesis.
Yeah, exactly. A long time ago. It was actually one of these cool things that was discovered in Xenopus in the frog as a maternally localized RNA. So in some
Oh,
animal eggs laid by the mother, there are some RNA molecules that get localized to one side of the embryo or one side of the egg. And so, from a long time ago, like decades ago, it was hypothesized that this gene, VEG1, has to have some kind of critical role in development because why would you segregate it to one side of the embryo if it wasn't to then define some property of that side? But all of the experiments that were done
huh.
couldn't reveal, like, there was no obvious function for it. And then during my PhD, CRISPR was developed and as a gene editing tool. And for the first time in zebrafish, we could actually knock out genes. And so I knocked out this gene in fish, and we had this really dramatic phenotype that looked so that the embryo was kind of messed up in the same way that a nodal mutant is messed up. And so we knew it must be involved in the same pathway. And so we gradually pulled on that thread and figured out the kind of
So
mechanism.
the veg one is in the egg, it's like to one side or whatever. Is that what you're saying?
In the frog egg, it is. It happens that in the fish egg, it's everywhere.
Oh, okay.
But we found out that it's actually inactive in the absence of nodal. So it's stuck inside the endoplasmic reticulum until nodal comes
Oh,
along and drags it out and forms a dimer, and then it can be active.
wow, that's fascinating.
Yeah.
I could, I don't want to pursue this now because we want to get to the
Yeah.
thing, but that's really cool. And because what I was thinking is by like if you, can you, how long can you go and then knock it out and still get the separation of the, or can you like decrease the expression of this protein at different time points from the whatever two cell stage onward and then figure out is it required like during the first cell division? Is that already determined somehow down the road what's going to be cell, what daughter cells will become
Mm.
mesoderm and what will become endoderm?
Actually, we don't have a way to control when we knock it out, but
Ah,
it is not required until it's a few hours old.
okay.
So, luckily, we took advantage of the fact that it's found in every cell and it doesn't do anything for the first few hours. And then if we knock it out everywhere, then we get this effect, strong effect. So we kind of could, yeah, figure out what it's doing from that.
And then does it have a role in the adult in some other function?
Yeah, so it does actually not in the well, I don't know about the adult, but
Okay.
later in development, the nodal pathway, after it's been used early in development to decide what's endoderm and mesoderm, later in development, it's used to decide what's your left side and your right side.
Oh,
So what's called left-right asymmetry. So it
yeah.
gets reused then,
Huh.
which is kind of cool. And there
Yeah.
were like hints that this is what's happening from mouse experiments, but I think we didn't. I mean, our lab was studying the nodal pathway and we didn't assume that it was involved at all. And then it turned out it had this key role all along, but it was kind of invisible, which was pretty cool.
Okay, so then you're working on zebrafish for your PhD, and then how did you get interested in octopus, squid, and cuttlefish?
Yeah, I think as I mentioned, like very early, I don't know, when I was a teenager, I realized I wanted to be a scientist and I was very dedicated to this idea. And I think the older I got, the more I realized that it's a very competitive career.
Yeah.
And I also realized that so many of the papers that so many of the discoveries we were making in our lab, other labs were making at the same time,
Uh,
which is good because then it means you have this immediate confirmation that
yeah.
your findings are real. But there was something a little unsatisfying about it to me that I felt like, what kind of original contribution do I make if someone's always making the same discovery in parallel? And so I felt like I want to try and find a new niche, do something different where I don't worry so much about what others are doing. And also, maybe I could be a little bit more creative. So I was trying to figure out what that would be. And I realized over the course of my PhD that a lot of the talks I was listening to, like other scientists coming to visit, that I got most excited about were people working on new model organisms, like weird creatures, and asking some kind of big question that maybe hasn't been asked before. Um, so like a lab working on navigate 3D navigation in bats, because navigation is normally studied in mice that only, move in 2 dimensions, or the origin of multicellularity in choanoflagellates, um, or the genetic basis of evolutionary change in pigeons. And these kinds of talks got me like really excited and made me kind of excited for paving my own way rather than fearful of paving my own way. So I kind of realized, okay, I think this is what I want to do, but I have to figure out what is the organism, what is the question, and so forth. Um, so in the penultimate year of my PhD, I went to the Marine Biological Laboratory, or the
Mhm.
MBL,
Yeah.
in Woods Hole, which is a famous place that people have been going to for over 100 years to do scientific courses. Um,
I taught, I can't remember, this is a long time ago, in
mm.
the 90s, I guess. I taught a course there this one summer.
Do you remember which one it was? One of the neurobiology courses,
Yeah,
maybe?
yeah, it was
Yeah.
neurobiology.
Yeah. Yeah, they
Yeah,
have
no,
two.
that's cool.
It's an amazing place because I mean, for most of the year, it's pretty quiet. There are year-round scientists. But in the summer, I mean,
Yeah.
people from all around the world just descend upon this place and it's just bubbling with scientific activity.
And then they
Uh,
have places for them to stay while they're there too.
yeah, exactly. You stay in these little cottages in the
Yeah.
woods. And yeah, for someone who's from London, lives in New York City, it's a pretty big difference, but it's really it's a very exciting place to be.
Yeah.
And I took what's called the embryology course. So it's a sort of developmental biology course. But I think unlike the other courses, it's more open-ended. You kind of march through the evolutionary tree, just trying different organisms every couple of days. And they give you suggestions for kinds of experiments you can do, but you're basically just set free in the lab to do whatever you want. And this is my favorite situation to be in. So I was just playing around with everything from tardigrades to butterflies
Huh.
to everything. But it's a 6 week course and I got 5.5 of the weeks through it. And I still I hadn't found the question and the organism. And then we had just a 10-minute talk they had a new manager of cephalopod operations. So octopus, cuttlefish, and squid are called cephalopods. And they just started they've been studying squid there, a local squid for I think over 100 years, but they were starting to culture tropical species at the MBL. And so the man who was doing that, Brett Grassi, he gave us just a 10-minute talk on cephalopod biology and the progress they've made towards culturing them. And I think just my whole life flashed before me in those 10 minutes. And I just
Wow.
knew immediately, like, this is what I've been looking for. Just there's so much amazing biology. And I think the hardest part for studying a new organism is how do you culture them? Like, how do you grow them in the lab? How do you get them to reproduce? And he and others had sort of solved a lot of those problems. So I felt like now is the time. And I haven't looked back.
Okay, but and then right. So then, but you didn't. Okay, so when you're looking for a postdoc, did you did you look at some labs that are already studying these cephalopods, or how did you end up in Richard Axel's lab? Because he was
No,
anybody from no,
there was no one already studying them. Yeah, I think with these situations, you have a choice of you could find a lab that's already studying them,
yeah,
but maybe the subject matter is different or the expertise, or you find the right scientific fit and try and bring the new project. So I mostly just applied to
I
labs that I thought were doing really cool science and thought about the type of question, um, and then I could bring the organism. So it took a little bit of convincing to Richard, but the reality of this as well is that cephalopods are extremely expensive, and so I did need a lab that had a lot of scientific funding.
uh,
So I'm very fortunate that he was excited and willing to do this because we've had to build an entire cephalopod facility and yeah, I can go into the lengths that we've gone to make this happen. It's a little bit insane at times, but I felt that we had a similar his lab traditionally is interested in olfaction, as you said, and sort of how does the brain create an internal representation of the olfactory world? And I think they just use olfaction more as an entry point to just understand how information is represented in the brain. It's not necessarily that the lab deeply cares about what we smell. And plus, they're using the mouse, which has evolved to interact and sense the world primarily through smell. So I argued to him that the cuttlefish, as I can go into, they instantaneously camouflage to their environment. And when they're camouflaging, they're basically reproducing what they see on their skin. So the brain is taking the visual world, creating some kind of representation, but then rather than just perceive the world, it then shows you its perception on the skin. And I'd argue that pretty much no other animal can do that, except maybe a human can describe what they perceive. So we're trying to take advantage of the fact that the animal is reporting its perception to learn about how the brain does that.
okay. So what's happening in the skin so that it can um, yeah, so that it can change color and
Reproduce the
pattern.
well.
They have to have some pigment somewhere.
Yeah, so the skin is covered in either thousands or millions of, you could kind of call them pixels. So they're these sacs of pigment, they're called chromatophores, and it's a flexible sac of pigment that's surrounded by radial muscles, and each of those muscles is innervated by a neuron that's in the brain. So the neurons in the brain project all the way to the skin without synapse, and then they control these muscles. And then the chromatophores come in different colors. So let's say the animal sees a brown background and it wants to turn brown. Then the signals in the brain, the neurons in the brain that control brown chromatophores, they will send the signal, activate the neuron. The neuron will contract the muscle, so the chromatophore suddenly expands to make a visible pixel. And then if you expand, I mean, all the brown chromatophores, the animal turns brown, but it can do more complex patterns and it can also make three-dimensional texture. So it happens in less than a second. It's incredibly fast, and I think it's the fastest known animal color change on the planet.
Yeah, I mean like chameleons are yeah, there's a lot of animals that like they can kind of have a uniform color change, but they don't have any patterning and it doesn't happen very quickly. I was going to ask you, so and then it should I guess it's the same in the cuttlefish and squid and Uh, is the neurotransmitter at the neuromuscular junction, the neurotransmitter that causes the muscles in these chromatophores to contract, is it glutamate?
Yes, it's glutamate.
Yeah, the most important neurotransmitter.
Oh yeah.
I actually wrote a book when I retired because my PhD, not my PhD, my postdoc, I discovered that glutamate controls formation of synapses during development of the hippocampus,
Mhm.
and then did all sorts of work on glutamate in relation to brain aging and neurodegenerative disorders because you can kill neurons. So I wrote a book that's called Sculptor and Destroyer, and it's all about glutamate. And
That's a good title.
it's role in development, plasticity, and then degenerative condition.
Yeah.
Okay, so it excites the muscle, get calcium influx, they contract, and
And
all
then the
right,
chromatophore expands.
yeah.
Mhm.
All right, so where are the cell bodies of these uh, neurons that are innervating the chromatophores? Where in the brain?
They're in a region called the chromatophore lobe.
Oh,
There's an
okay.
anterior one that controls the head, and the posterior one controls the body. So, yeah, it's nicely demarcated in one particular region.
You had a paper on in Current Biology, which is a really good journal, on brain atlas for the camouflaging of the dwarf cuttlefish, which you have in your marine room, I guess.
Hmm.
And right, and when I looked at there's kind of an overview of different, but like the optic lobes are huge,
Hmm.
right? And what's it is the ret where's the actual so you have an eye and then that projects to the optic lobe or
Yeah, so they have two eyes kind of on either side of their head that seem to have a really wide field of view, almost 360 degrees. And then the neurons from the retina project directly to the optic lobes, which are these massive, they're like 75% the volume of the
yeah,
brain.
it's
And
amazing.
then between the optic lobes is a central brain. And then their esophagus passes through the middle of their brain because invertebrates are weird.
Yeah.
Um,
And then
yeah.
the optic lobe, uh, right, what's the organization? So in usually in kind of these brain regions that are processing information that's coming in, you have like a core circuitry of I'm thinking about mammals now. Core
Mhm.
circuitry of excitatory neurons, and then there's inhibitory neurons that kind of prevent things from getting out of control, and they play an important role. What is it known what the circuitry within the optic lobe is like in terms of what neurotransmitters neurons use and its organization?
Yeah, not much in general, as you'll see. We don't know much about much.
And that's
Um,
good. That's good for you.
yeah, exactly. There are lots of open questions. I mean, what's cool about the cephalopod brain is already in the 1900s, Santiago Ramon y Cajal was already interested in the anatomy. And then these scientists, J.Z. Young, B.B. Boycott, um, and J.B. Messenger were kind of figuring out a lot of the anatomy. So we do know the basic anatomy, which is that the outside of the optic lobes have a couple of layers around them, which has been called the deep retina because they actually seem to be more like the vertebrate retina.
Oh,
Um,
interesting.
because the retina itself in the eye is just a single layer of cells. But then after you get from those outer layers, it gets really weird. So you have these things called cell islands where all the cell bodies form what look like islands with
Hmm.
all of the axons or neuropil in between. But a group of scientists imaged it in 3D and realized that they're not actually islands. They're all connected like branches of a tree. So it's sort of like you have a trunk in the middle connecting to the central brain, and then you have all these branches going throughout the optic lobes. And basically, we know very little about how this structure allows them to do visual processing. But it's at least evident, I think, or it's the fact that 75% of their brain is dedicated to visual processing suggests they're very visual animals, which is consistent with what we see in their behavior.
And right, so in your you're studying embryogenesis in the zebrafish embryo, and
Hmm.
then so one of the I guess I'm just kind of guessing from looking at what you've published that one of the first things you did when you got to um, New York, New York City, was to look at embryonic development in and of the of this cuttlefish. Is that
Yeah, I
right?
mean, really the goal of my postdoc and maybe my whole career is to record or image neural activity in a cuttlefish while it's camouflaging in a virtual reality so that we can show it a stimulus like polka dots. We can image neural activity to see how the brain's representing those polka dots. And then we image the skin and see how the brain transforms the visual input into this approximation on the skin. And by seeing this transformation of the world into a pattern on the skin, we can hopefully uncover how the brain is doing that, like how brains represent information, how brains transform information. But to get to this ultimate experiment, there are so many tools that we have to develop to do it. And when I started, like the MBL that I was talking about, the Marine Biological Laboratory, they already knew how to grow the species I'm working on, called the dwarf cuttlefish, and how to get them to breed. But other than that, there was like not a single tool available. So I've been sort of systematically developing the tools to be able to perform that ultimate experiment. So we did look at what development looks like, but actually, even though I was a developmental biologist in a previous life, it wasn't just because we cared about development, because we're injecting the embryos and trying to make
Ah,
transgenic cuttlefish.
oh,
And once you start messing around with embryos, you need to know what normal development looks like.
oh,
So we kind of did that as a stepping stone to genetically engineering cuttlefish.
oh, cool. Okay, so yeah, so one thing you need, right? You said the idea is you can have so you have uh, a a chamber with water in it with
Hmm.
like screens, TV screens on each side, I guess six sides or whatever. And then the idea is you put some, so have you done this? That you have that and you can put a pattern on the screens and the animal will change its skin?
Yeah, so we don't actually use TV screens because we find that they react weirdly if like TV screens emit light. Plus they have like they polarize light and so forth. And the animals I
Huh.
think don't respond well to light, for instance, coming from underneath them. Because obviously in the wild, I mean, with
Just-
most creatures, light comes from above.
So you just put a picture out on this.
Well, we started with pictures and then we ended up with e-ink, which is the technology in Kindles. Because this
Oh,
like
ah,
Kindles were developed to not emit light and to be gentle on your eyes, and
yeah.
they actually use true pigments. So they have black and white true pigments that are charged and they come to the surface of the screen to make different patterns, which is why it looks a bit like paper because it's actual pigment and not light. And so we use these screens on, yeah, five sides of the tank, all four sides and underneath, and then we emit light from above and the animals camouflage sometimes. I think one of the discoveries I've made of moving into neuroscience is that animal behavior is very complicated and animals mostly do whatever the hell they want, not what you want. But we definitely, I don't know, let's say 10% of the time, they do remarkably good camouflage to a black and white flat screen, which is pretty amazing because obviously it's very different from their natural habitat.
Okay, and is it right? So you're taking the cuttlefish. Usually, when you handle an animal, right, like a human, like you move it into this chamber, that's a stress.
Mm.
Um, oh, so the camouflage, are they changing of the skin color and texture and stuff? It's not just for like hiding and, hiding and then whatever prey comes by and they grab it. They also use it for like, uh, al- what would you say? Alerting or telling other animals, I guess, of the same species or another one, hey, get away or stuff like that, right? So anyway, is there a role for stress in whether how the animal responds to the artificial pattern? Maybe if they get more adapted, they're more likely to, I don't know.
Yeah, no, it's a sort of complicated topic because I think, I mean, the first thing is, as I said, their skin is controlled directly by the brain. and they're always changing their skin pattern in some way. Like if you look up
Mm.
close to a cuttlefish, the chromatophores are kind of twinkling all the
Mm.
time. So it's very dynamic. It's not like it's kind of off and then only gets turned on in certain circumstances. Um, and with that said, they sometimes use it for camouflage. Other times they're not camouflaging, and we don't really know what they're thinking or what the goal is here. Other times, if they're actually quite stressed or fearful, I think they, rather than use camouflage, they produce what's called a dymatic pattern, where they kind of go really white or pale, and they make these false eye spots on their skin, which I think is a kind
Yeah.
of like when octopus do it, they also stretch their bodies really big. So it's kind of like a last resort, like I'm so scary, you don't want to mess with me.
Yeah.
Um, and then they also have this whole set of amazing social skin patterns that they use to communicate with each other. So, and then sometimes when they're sleeping, they also make skin patterns, dynamic patterns. So basically, I think what's really exciting about these organisms is because their skin is connected to the brain, you have this physical readout of all different processes that are happening in the brain, not only camouflage. But in terms of like whether they have to be stressed to camouflage, the annoying thing is we can't if the animal is like in the digital tank and is not camouflaging, if we like open the curtain, maybe for a split second, they'll suddenly do a much better camouflage. But if we try and do that again, they won't fall for it. We haven't figured out a way to have them constantly in a state of feeling that they need to camouflage. But
There's
also if we
there's so many variables that are unknown, right? I mean, is it known if there's any circadian effects, or are there male female differences or that kind of thing?
Yeah, so there are definitely circadian differences in their activity. So we built this coral reef tank in our lab and we put some sort of security cameras on it because we could just stream it 24/7. It was on my website. We've temporarily taken it down because we need to like remake the tank a bit. But we saw that at night, like we considered them diurnal animals because when we walk through the facility, they're generally awake and bobbing around. But once we looked at night, we saw they were crazy active at night.
Ah,
And we actually just went to the Philippines in November to study them in the wild, and they're definitely nocturnal animals there. But so there's definitely a difference in activity level by day and by night. I think to know if their skin patterns are different, we need to be able to like really quantify them. And this is one of the biggest challenges is like, how do you quantify a really complex image, basically?
Do you feed them? How often do you feed them?
Oh, this is a very sensitive topic. They have to be fed live shrimp 3 times a day. So they're very needy animals.
Um, right. So my lab did a lot of work on fasting, intermittent fasting,
Mm,
mostly in mice and rats. This was in the 90s, and it actually was our lab's early work that led to this. Now, intermittent fasting is a big thing.
mm,
But what if you deprive the animals of food for a couple days under conditions when it's really important that everything's working right for them to get food,
mm,
and then see if you get under that condition, they're more likely to generate a pattern. Just thinking from like an evolutionary perspective, if they're hungry, then they have to focus on hiding and getting food or something like that.
mm. Yeah, it's true. We tried once an experiment where we did some fasting and then we were trying to train the cuttlefish to change a certain skin pattern to then get a shrimp. But it was quite complicated and took like weeks of testing. So I think maybe we need to do something a bit simpler, like you're suggesting, to see
Yeah.
whether or not we can, yeah, coax them to do what we want.
Okay. All right. So there was a paper that I think it was another lab was the main lab, but you're an author on it on making albino, and I don't know
Squid,
if it was a
yeah,
cuttlefish, it was a squid.
was squid, yeah.
And So in the albino squid, are the chromatophores still there? What are you eliminating? What,
Yeah, that was, I mean, I think the reason to make the albino squid was just a proof of principle that CRISPR works. Kind of
uh,
like when people have made albino, I think lizards and fish and everything. Just because then if you successfully got a mutation, you can immediately see whether or not you did or not. So for that, those experiments, they were targeting an enzyme that's used to convert a molecule into pigment. So I think the chromatophores are still there, but they lack the pigment.
okay.
You can't see them. But the problem is they also need that pigment in their eyes, so then they're blind, and so it's quite complicated.
Oh, wait. No, so here's the thing. So what
Hmm.
you need to do, right, is get like a calcium indicator into the muscle cells that control the chromatophores. So that's then you have a readout. You have a you can image really well because there's no pigment, right? But you can know when the muscles are contracting, which is equivalent to the chromatophore.
Yeah, I think the method that we're planning to do is we're trying to put GCAMP, this calcium indicator, into the neurons, and then kind of like mice, put actually a lens inside the brain
Ah,
and have a mini microscope
yeah.
so that we can image the neurons that control the skin and then also film the skin and see what the output is. Um, but it's
And you can, all right, so then you'll be like, you'll have two recordings, the neuron and that pattern like in real time or whatever.
yeah,
But
exactly.
you could also use optogenetics, right? So you could stimulate uh, the neurons in the brain and see how it affects
Exactly. Yeah,
that.
that would be the next step, which hopefully we'll get to, but making transgenic cuttlefish is extremely difficult.
Uh-oh.
Uh-oh, yeah.
And you
I
can't,
think we're close, but
one downside, so we'd use a lot of, and mice can use viral vectors to put, you can even, with AAV, they have some different like serotypes or whatever, so you can target the AAV with whatever gene you want to express, we did a lot of siRNA work to knock down expression using RNA interference of genes, like only in certain neurons and stuff.
yeah, I
So
think
there are some disadvantages to the invertebrates when there's certain techniques that seem are tools that haven't been developed yet.
yeah, exactly. Yeah, yeah, viral vectors would be a nice alternative. So we've collaborated with a couple of people to try and develop viruses. Like the kind of vanilla viruses that work in mice and other organisms don't work in. They don't seem to infect cephalopod cells.
Yeah.
But actually on Sunday I injected a new group of viruses from our collaborator Kim Ratola. So maybe this will be the magical group. She did some interesting serotyping so we can see if maybe these will infect cephalopods. But unfortunately, most things don't work and they take a huge amount of work to get them to work. So this is the big downside of working on a new model organism.
Yeah. Well, it's a dumb, but like you said, I think it was a good decision. You made this decision and it's hard work and it's going to take a long time, right? This your
Mm.
career, but hurdles will be overcome. And yeah.
Yeah, I think
Okay.
the payoff will be worth it.
All right. So you have a number of what you're mentoring graduate students, some graduate students, maybe some undergraduates. You have a few technical people. So you're essentially kind of independent. Um, so what, so at this stage, h- I guess this is not a too specific a question. So you got a great environment there. Are you going to continue with that? Is it likely that then maybe you can somehow transition into a, a tenor track position? Or because right now you're kind of you're independent, but you're not independent?
Yeah, I'm still technically a
Yeah,
postdoctoral fellow. I will go on the job market in like 1 to 2 years, which is it's pretty long postdoc. I've been a postdoc for 6.5 years. So this this project, as I mentioned, because we have to build everything from the ground
yeah,
up, everything from scratch. It's been a long journey. Um, so I think, yeah, I would normally, and maybe I would normally go on the job market this fall. But things are pretty bad in the
yeah,
world of science, and I'm hopefully close to making this transgenic cuttlefish, which has been the hardest thing of all. And I want to have that full toolkit before I start my own lab. So yeah, maybe in a year, just over a year, I will go on the job market and then officially start my own lab. But I'm very lucky to be in Richard's lab because he provides all of this support while I also get to have a lot of independence, which is
yeah.
amazing.
And okay, so one obvious, you know, kind of, yeah, it's very obvious implication of your work has to do with the military, or maybe not
Mm.
now with all this AI stuff and autonomous vehicles, but um, what kind of oh, okay, so there's kind of something we didn't talk about, like what computations might be going on in the brain to translate the input coming from the retina to the changes in the skin, right? Obviously, there's these neural circuits involved, but in a general sense, it's not, the animal isn't, you know, accurately reproducing the actual pattern in detail, right? But it's
Mm.
approximating it, or it looks like it enough that they can blend into a background. What have you must thought a lot about what's going on there in terms of the computations.
Yeah, yeah. So, as you said, what's interesting about these animals is they don't perfectly recreate the world pixel for pixel. I mean, I think it would be pretty amazing if they did, but in a way, you could argue it's more impressive that they're able to create an approximation that is actually extremely effective at camouflaging them. But also, what's kind of amazing about it is they've presumably evolved to camouflage from predators and prey. So, like other cephalopods, fish, shrimp. So, all these very different visual systems, but then also they successfully camouflage from us, like humans. So, it's kind of amazing that whatever this computation is they're doing, it it has evolved to kind of trick a huge range of visual systems in terms of how they're doing it. So I'm not a computational neuroscientist by training, but one of the graduate students working with us, Erica Shook, she's a theorist, and we've been working with Larry Abbott, who's a theorist at Columbia. And Erica has been thinking about this problem in terms of something called visual texture. So, in general, you could say that our eyes, our visual systems detect two types of things. They detect objects, like a water bottle, and then they detect textures, like leaves. And what a texture is or a visual texture is it's a kind of image with repeating units. It's spatially homogeneous, and there's some random variation. So, like leaves or grass or sand or corals,
Mhm.
it's sort of spatially homogeneous. They're repeating features, but it's a bit random. And we know that the primate brain, like ours, encodes what's called visual texture as well as encoding objects. But Erica thought that maybe the cephalopod brain is specifically has evolved to detect visual textures because we know that the underwater environment has a lot of visual textures. They said like seaweed, sands, corals. But also, if you look at cephalopod camouflage patterns, they are very textural as well. And so you could imagine that the brain has specifically evolved to represent this kind of information. And what's really cool about visual textures is a scientist at NYU called Eero Simoncelli. Him and Javier Portilla have shown that you can basically take a visual texture, you can gradually figure out, like iteratively filter this image to extract a bunch of statistics. And using just these statistics, just a group of numbers, you can recreate that texture or an approximation of that texture. So you couldn't do this with an object, but with something that's spatially homogeneous, has these repeating units, there's random variation. You can extract just a set of numbers and then recreate an approximation. And
Huh.
so that's what the cuttlefish does. It takes an image, it must extract some kind of low dimensional representation, but then it recreates an approximation of the input. And there's a computational model called a variational autoencoder that does this. So Erica and Larry and us now, we're convinced, think that probably the cephalopod brain is functioning a bit like a variational autoencoder. And so it's specifically detecting visual textures. It's then encoding them in some small set of summary statistics, and then it's sort of decoding that to recreate an approximation of the world on the skin.
Huh.
Um, so we have a model,
Yeah.
and of course, well, and Eric is now going to try and build an actual computational model of what, so we can start forming predictions. And then as soon as I've made this transgenic cuttlefish, so we can image neural activity in the brain, we can see if we can find this kind of representation of those
Huh.
statistics.
Okay, that's cool.
Yeah.
Yeah. I hope that goes quickly getting the transgenic
Thanks.
cuttlefish.
I hope so too.
What was I going to say? What about the, I don't know if these are kind of sloppy experiments compared to what you're planning, but like, and this is also a problem, somewhat of a problem with invertebrates, is it pharmacology? In other
Mm.
words, what about blocking receptors for certain neurotransmitters or adding agonists of certain neurotransmitter receptors and seeing how that affect. I guess once you get the transgenic animals, then it might be better to wait. Then you can actually look at how they're affecting the activity. In other words, can you stop a cuttlefish from having camouflage in response to a background by blocking whatever acetylcholine receptors or GABA or glutamate?
Yeah, I think we could try. Other than knowing that the motor neurons that innervate the skin are glutamatergic, we sort of know that lots of different neurotransmitters are expressed across lots of the brain, but we don't know anything much more specific than that. So I
Yeah,
think it might be difficult to do a very targeted
yeah.
um, interference in the brain. As with lots of these things, yeah, we come across the issue of we maybe don't know enough to even start, and then we also don't know if any of these reagents work in cephalopods. So
So you need to connectome, right? That like in Drosophila recently they got the whole
yeah, the entire
connectome
brain.
done. And that those methods, is there someone at Columbia doing that kind of stuff? With
Um,
EM, like you do these serial electron microscope sections through the
Yeah, I think, well, actually, Gilles Laurent, who's a group leader at the Max Planck in Frankfurt, his lab also works on cuttlefish on a
huh,
different species from us, but on camouflage. And I believe they've started a collaboration with people at Genelia to do a
ah,
connectome. But the thing is that the cuttlefish brain, I mean, we work on the smallest cuttlefish species, and their brain is the same volume as a mouse brain. And whereas
oh
Gilles' lab,
really?
yeah,
Well,
works
how
on a
big
much
is the cuttlefish?
it's like this big. Um,
Ah,
so their body is up to like 70 millimeters. So the whole body is about, yeah, 80 millimeters, 8 centimeters. Yeah. I know
okay.
it's like you kind of they're small. The
Yeah.
species that other most of the other labs that work on cuttlefish work on what's called the common cuttlefish or the European cuttlefish, and that grows to be like this big, which we didn't want to work on that species because we want to breed them, make transgenics, etc. But anyway, those in the Laurent lab is starting to do a connectome with Genelia, but because the brain is so big, they're doing like a tiny chunk of the optic lobe.
Oh yeah.
Um, so I think, yeah, probably to gain the most from that, you'd want to have a connectome for an entire brain, and I think the
That's a lot of work.
technology is there yet.
Yeah. Okay. Ah, but there's this the method. Oh my gosh, we never used it, but the method where you fix an entire brain in fixative, like whatever
hmm
paraformaldehyde, and then you get rid of all the lipids,
Clearing, like brain clearing.
brain clearing, and then
Yeah.
in mammals there's a lot of good antibodies, but that's another problem with invertebrates, right?
Yeah.
Uh, no, but you can express, you should be able to have animals expressing like the rainbow type of thing that
I mean,
Jeff
we can't,
Lichtman developed or something like
yeah,
that.
I think for that you need transgenics, but so a graduate student in the lab, Thomas Barlow, he's been clearing brains and doing some nice anatomy, but
Huh.
as you said, only some antibodies will work. So we've tried a few and a small percentage of them work. We've done tracing, which is nice. So we can inject a
Ah,
dye into one part of the brain, then see these like
ah,
beautiful projections to another part.
yeah.
Um, and we've sort of confirmed that the connectivity of the camouflage circuit is what we expected. Um,
Can you inject into the muscle cells innervating the chromatophore?
oh, I wish. Yeah, so we would
They're
love
too
to.
small.
Um, those are too small. So to inject into the skin, in theory, it sounds great. And then you look at the skin, you think, oh my gosh. Um,
Whoops. Uh-oh.
did you lose me?
No, I don't know what happened. It must have
Okay.
been some alien interference or something.
Um, yeah, we've been trying to inject into the brain and trace all the way to the skin, but at least, I mean, there are
Ah.
so many things to do. We haven't dedicated enough time to it, but it never got all the way to the skin. I mean,
But
it's an immense distance. So
these are all good projects for graduate students.
yeah, yeah, I
Yeah.
think maybe the danger of a new model organism like this is there are so many things you can do,
Yeah.
and so how do you restrict yourself to the most important things? Or
Yeah. Yeah.
it's a
Okay.
difficult balance.
All right. So anyway, right. So you've talked about a lot about what you plan on doing and a lot of good things to do, a lot of technical hurdles to overcome. Down the road, what, not that your work has to have any direct implications to uh, humans, but can you kind of imagine
Mm.
something that can be learned from studying this camouflaging mechanism that is relevant?
Yeah, I would say that first of all, I would say that of all the project, like working on a new system, like this project has been my dream, and everything has been wilder than or beyond my wildest imagination. But the one thing that I wish it had was a really clear connection to human health. I think some new model organisms like mosquitoes, or you could argue they're not that new anymore, but all the development of tools, like you have this really clear connection for how you could impact,
Ah, yeah.
human health. And I think cuttlefish
But you don't
camouflage,
know, like, so Tessa, like the worms,
yeah.
like there's so many genes that were discovered in the worms that turned out to be like relevant to cancers, like BCL2 family members, or
Mm.
in yeast, I mentioned autophagy, which is going on. But you may just, I think it's likely that when you get so that you can start manipulating genes, you're going to discover some gene that has a homologue in mammals and its function isn't known in mammals and like that. I don't know.
Yeah, I think that's what I'm hoping is that if you try to do the most creative stuff you can and do it as rigorously as possible,
Yeah.
and if you use an organism like cephalopods that are just have created so much biological innovation, things will come out that will impact the world in some positive way. And sometimes you need to just have an open mind and try and discover, and it will happen. So I mean, I would say with camouflage, the hope is more that we just get some fundamental insight into how brains represent information, and then
Yeah.
long term, we find that there is some conserved principle with like how human brains represent information. Um, I mentioned that cephalopods also do these amazing social skin patterns.
Hmm.
So this is something we just started studying quite seriously recently. Like if one cuttlefish is very angry, it turns jet black and puts these little white specks on its skin, and the other one does it too, and they flare at each other. And then one does this leopard print pattern, and then they do these waves on the skin. And you just watch these like incredible bouts and this sort of visual language occurring between the two
Yeah.
animals. And you could imagine that somewhere down the line, we could, given that we can see the communication between animals, we can see the moment an animal becomes dominant to another animal, that maybe we could then investigate, when there are issues with social communication. Maybe we could get
Huh.
some insight into the brain
Interesting.
and when animals don't pick up on social cues and what implications that could have for humans. Um, but then there are other cool things like there's a squid species, bobtail squid, the one that the CRISPR paper was done in.
Huh.
Um, they have this skin glue that they secrete to hold, like rather than do really dynamic camouflage to hide, they secrete some kind of mucus glue and they glue little rocks and sand onto their skin.
Ah,
But if you kind of scare them, even though all of this stuff is stuck to their skin, just in an instant, they can just release the whole puff and swim away. So they have like an anti-glue. And I'm very
huh.
interested in the idea that we
So
could,
they're not just like shaking it off. There's some,
no, there's some release because
huh.
it like split second, the whole thing just falls off as if it was never there. And so, like, perhaps we could, purify whatever this compound is that they've evolved. And maybe you could use this in, heart surgery one day when you need to glue something
Huh.
temporarily and then you
Yeah.
suddenly release it just when you need to. Or I think there are various innovations. I mean, like the suction cups that we use to stick things to walls, those were inspired by cephalopods because on their arms they have these suckers.
Yeah.
Um, and they do so many weird and wonderful things. So I'm sort of confident that in the long run, we will
Yeah.
make discoveries that have an impact for human health, but I don't know how much I can predict what they will be.
Okay. Well, I hope I'm still around to see this. Um, what's the last thing? Global climate change. And some of these, I don't know if the cuttlefish it's true, but some of these cephalopods live in coral reefs, and I know there's loss of coral reefs. There must be some work going on, or maybe not, on how climate change and warming of the oceans and affects these creatures.
Yeah, yeah, a lot of the species, as you said, live on coral reefs. And like we went to the Philippines in November to go diving with them, and it was amazing seeing them doing the real in the wild, doing their real camouflage in the real environment. And the coral reefs were beautiful, but there was also signs of anthropogenic change. There was trash everywhere. And
Hmm.
I think, I mean, a lot of octopus actually successfully live in human like trash. And
Plastic stuff, probably.
yeah, like in jars and cans. I don't remember the statistics, but I believe there is evidence that they are doing pretty well in the face of climate change.
Hmm.
Unlike a lot of fish species, they are sort of evolving somehow, or they are adapting, I
Interesting.
should say. Um, but one of the interesting things about cephalopods is they have a really high rate of RNA editing. I don't know if you know about this, but so there are these enzymes that mammals have too, called ADARs, and they can basically take the sequence of an RNA and they can convert some of the A's into uridine that gets read off as a G. So basically, they can change a couple of letters in an RNA sequence that therefore changes the protein sequence.
Huh.
And the reason this is sort of interesting is it means from a single gene, rather than one gene making one protein, you could make potentially like dozens of proteins from the same gene because in different places you tweak the sequence to then make different protein
Hmm.
sequences. And I can't remember the statistics, but Mammals have like a few sites around the genome that they can edit, but cephalopods have huge numbers. Like they're just all across their genome, their transcriptome, they're editing sequences.
Interesting.
And there's some data to suggest these might have functions, but there's a kind of exciting idea that maybe they are basically adapting them. They're more adaptable to changing climates and changing ocean
Oh,
temperatures and so forth because they can very quickly
right.
make a different protein, like a different channel that has different properties. And one of the cool things about cephalopods is a lot of them do what are called diel migrations. So during the day, they're really deep in the ocean. And then at night, they swim upwards like hundreds of meters. And then at night, in the day, they go down again. So the temperature is changing, the pressure is changing. There are all these
Oh,
um, environmental shifts that are happening over the course of minutes or hours. And perhaps their ability to edit their RNA allows them to rapidly adapt to these changing conditions. And so then maybe they can also use this same system to adapt to changing oceans and in the face of climate change. But
I
most of
wonder.
that is just theory.
My wife worked on zooplankton in the Chesapeake Bay for a while, and they do the kind of the same thing, right? They
Mm,
in the day they're down low where it's cooler, and then they come up at night. And but I don't know how much has been studied at the molecular level in the zooplankton. But
yeah.
the cephalopods, well, somehow cephalopods are getting energy from zooplankton, probably.
Well,
They're up the food chain, but
yeah,
things like
ultimately.
the filter feeders uh, consume the zooplankton, and then those are consumed, like that.
Yeah, yeah.
Yeah.
Yeah, definitely. Yeah, and actually when we were diving with the cuttlefish in the wild, we did a cool dive called a blackwater dive where you go at night and you park your boat over a really deep part of the ocean and then you drop this line of lights into the water and you leave the lights to stew and basically, over the course of about 30 minutes, all the animals that are doing that dial migration and zooplankton, as well as all kinds of other organisms, they come up from the depths and go towards the light. And then you jump in the water and you go down and you just have a flashlight. And so you try to just float. It's quite technical because you have to maintain your buoyancy because you have nothing, you don't have like the bottom of the ocean to know how deep you are. And if you go too deep, you can get narcosis. But you have a flashlight and you hold it up to the water in front of you. And it's probably one of the most magical things I've ever done in my life because you see everything from like these tiny organisms to jellyfish and cephalopods and just these minuscule organisms that are like moving in the ocean and going towards the
Wow.
light. And then what's interesting is most of them, if you shine a light on them, their kind of predatory response is to start going down. And so you're there just watching this organism, and then you feel your wrist buzzing because your dive computer is like you're plunging down. So you're like, oh gosh,
Oh,
that because you just follow the organism. But it's like being in outer space. It's just amazing.
so you must have had experience diving before you went to the Philippines.
I had some experience, but I got most of my experience being out there. We
But
did,
you had someone with you so that
yeah, exactly.
you
We
didn't
had,
die.
yeah, yeah, we had dive masters and stuff there, but
Okay.
it was a little scary, but also fascinating.
Yeah, that's cool.
Yeah.
Okay, Tessa, I'll let you get back to the cuttlefish.
Yeah, well, thank you very much. This is really fun.
Yeah. Bye.
Okay, bye.